On the other side of the coin, omega-3 oils will be anti-inflammatory. And this is what we hear: eat fish oil or green leafy vegetables or flaxseed oil. And it actually will, aside from the fact that N3 or omega-3 fatty acids, when they break down when they're acted on by enzymes in the metabolic process. They break down into anti-inflammatory eicosanoids. They also influence gene expression. They also dampen down the expression of inflammatory genes. And then on and on. We see that resveratrol, actually down-regulates inflammatory genes, so we want to drink red wine, except we'd have to drink cases and cases of red wine really to have the effect, but it's still a good idea to have a glass of red wine, or at least we can tell ourselves that. But when you look at a meta-analysis, as this scientist has, of the nutrigenomic studies, the effect of one nutrient on specific genes, only 1% of them actually triplicate, which means that the problems that we have cannot all be predicted by our genes. That 70% is less the accepted estimate of illness is determined not by our genes but by our lifestyle. So this is a very well-known case and very good illustration of that point. The Pima Indians, who were indigenous to the plains in Mexico, and they had an agricultural society, many of whom were transported to reservations in Arizona. Now, they had the same genes, the ones who stayed in Mexico, as did the ones who were transported to the United States and lived on reservations. But the ones who were transported to the U.S. adopted the U.S. diet. They all became overweight and diabetic. And all of the Pimas who stayed in their indigenous area, eating their indigenous diet and being active, remained healthy and lean. So there's a good illustration of that. And this is expressed by the CEO of the private company that was the counterpart to the U.S. government in terms of mapping out the human genome. And this quote was made by him at the press release when the mapping of the genome the completion of that project was announced. Trey Ventnor, he says, we're not hardwired. The environment acting on these biological stuffs may be key in making us what we are. So even he, who had the company Affymetrix, where he would sell products determining gene expression, said, well, hold on. The fact is, really, that's not telling all the story. So now that I've I think we've gotten to the point where we see that food affects us in very complex ways. It's individual, and it's somewhat based on our genetics. It's very much based on our lifestyle. I want to look at a little bit, again, of the differences between the different types of adverse reactions we can have to food. And as I mentioned, the classical response to food allergy involves a specific branch of the immune system, which means going after specific pathogens, either by producing a specific antibody or a killer T cell has the receptor that recognizes a specific pathogen. And that's related to our genes, but it's also modified by our environment. We can actually rearrange our genes that produce antibodies. Symptom onset is immediate. Why? Because we're dealing with an evolutionary mechanism that is aimed at protecting us from worms, and they're big. And the reaction has to be pretty dramatic, in order for a small cell or a group of small cells to kill a big worm. So it produces dramatic symptoms, and therefore we know it. It was discovered in 1967 that the trigger was a specific antibody of this isotype, IgE, one of the 5 osteotypes. And it involved primarily these cells, basophils in the blood and mast cells in the tissue, that contain granules that release histamine and others. And you can test for that with immunological tests that measure allergen-specific antibodies, or actually putting the test substance into the skin, scratching the skin, and then if the antibodies are present, it will interact with this substance and sensitize the cells that are in the skin. And they also are lining other parts of the body, but they tend to be on the surface. The mast cells are. Intolerance is a bit different. The symptoms are delayed, and it could be that the symptoms are delayed by even more than a day or two. It's the innate branch of the immune system. It is also based on, for example, if we've not, a good example is many Asians do not tolerate milk. Why? They haven't had experience with milk for countless generations, as have Northern Europeans. So they have lactase deficiencies. It's very much based on the integrity of the gut. We hear about leaky gut as being a factor in many autoimmune diseases, and it is. And I want to spend just a few minutes going over a little bit of the mechanics of that. What can cause leaky gut? Allergy, intolerance, stress, antibiotics, parasitic conditions, combinations of the above, infections with yeast, candida, so forth. And then we have macromolecules of food particles coming into the circulation that are not fully digested, and then they trigger immune responses. The gut's interesting. There are more neurons. The gut's the only organ that you can sever from the brain and it'll still function. Don't try that at home. I'll take my word for it, but it is. People call it this. There's a book entitled The Second Brain, which is very, very interesting. 95% of the serotonin that is consumed in the body is consumed and produced. Right? It's used as a signaling molecule between white cells. People who have inflammatory gut often develop serotonin deficit and they get depressed, and they try and compensate for that by eating sugar or high glycemic index foods. Why? Because it induces an insulin spike, it drives branched-chain amino acids into the tissue, and the precursor of serotonin, tryptophan, is then having less competition across the blood-brain barrier, enter the brain, and be transformed into serotonin. So quite often you see people with food intolerance, they're tired for other reasons, and they're possibly lacking in serotonin. They like a lot of sugary foods, a lot of sweets, they crave it. Also, how well we transform the foods that we eat into non-toxic forms. All produce, I mean, you wouldn't know it today because we spray them with 50 or 60 pesticides. But up until times of commercial agriculture, which is recently coinciding with the end of the Second World War, when the Americans and the Germans had excess warfare chemicals, and they didn't know what to do with it, and they said, let's sell it to the farmers. But before that, we didn't use pesticides. So, how in the world do these plants actually survive for all these millions of years without these pesticides? Amazing. Yeah, but they can do it. Well, they have their own natural defenses and to resist pesticides. They also contain chemicals that induce bowel movements. Fruit does, we know that. Why? Because it's an evolutionary survival tactic. And the squirrel comes and eats the fruit and scurries away and then poops out the seeds 50 feet. It's better than if the seeds just drop straight down to the ground. The species has much better chance of survival. Now, if we're not used to that chemical, that can cause some irritation in our bowel. Or if, for example, we're lacking in some cofactor that the liver requires for normal biotransformation of that chemical into something non-toxic, it can become a problem that manifests through the GI system or elsewhere. So again, it gets a little bit more complex. And if we're eating foods that are grown artificially, they have less nutrients. So we have less nutrients available for that. And that results in enzyme deficiencies. It's interesting that the human lymphocyte, sort of the commander cell of the human immune system cells, contains all the enzymes and substrates that are in the liver for biotransformation. So what happens is, after the liver doesn't properly degrade some potentially toxic chemical, the immune system will. And that will result in some kind of bystander tissue effect. Started to speak about the gut. This is just a small sampling of how real critical. There's 100 million cells live in the gut, not our own origin. These are other organisms, microorganisms. They weigh about 2 or so pounds, maybe a little bit more. Approximately 1,000 different species. And the aggregate of that genome is 100 times greater than our own genes. And it has various effects on detoxification, gene expression, activation of PPAR receptors, which impact inflammation, how we absorb sugar, how we synthesize vitamins, many different things. What's come out recently is that certainly if we have a lot of antibiotics, or maybe even a little antibiotics, we inadvertently kill some of the bacteria, the eubacteria that would be good for us, and we disrupt things. And then other bacteria take over, and we get a dysbiosis, and that could impact our digestion, and so forth. What we eat also impacts the expression of the living organisms in our gut. There's a study produced at the end of last year from the University of Florence, where they looked at the gut flora of children in a town in Italy where they ate a typical modern Italian diet with lots of pasta and refined foods versus that of village African children. And they found that the gut profiles were totally different. And the African children were dominated by a class called Firmicutes. And those are the types of microorganisms that we see expressed in people who are also lean. And the industrially fed children expressed predominantly Bacteroides, which is characteristic of the gut flora of people who are actually overweight. So, very important thing to look at the gut. And it's a two-way traffic here. What we eat and how we digest it is certainly impacted by the health of the gut. And the health of the gut, likewise, is impacted by what we eat. It gets a little more complicated. This is an illustration, actually, I took from Alessio, Dr. Alessio from University of Maryland, who's been working Crohn's for a long time. That's one disease that is related to our genetic expression and also our exposures. Essentially, gluten, these are gluten molecules right here, intermingle with the enterocytes lining the lumen of the gut. And they'll elicit, if there's an allergy, a molecule called zonulin that loosens the tight junctions. That's where we get leaky gut. And then you get the gluten molecules coming in. They bind with something produced by the damaged enterocytes, tissue transglutaminase. It becomes immunogenic, gets taken up by an antigen-presenting cell, presented to a T lymphocyte, and then triggers more immune responses, either antibody responses from B cells or the T cells actually further destroy the lining of the gut. But it only happens, none of us really digest this gluten actually. It's very interesting. It only happens in people with a certain marker at this stage that at this stage, an MHC molecule called DQ2 or DQ8. A group of scientists at Stanford some years ago looked at some people with celiac disease, and 14 of them versus a control group that didn't have celiac disease. None of the patients were able to digest the gluten. It's a very tough protein to break down. But it didn't really bother most people. Only those people who had this expression of this major histocompatibility complex, which is how the immune system gets activated from an antigen-presenting cell through interaction with that one, which is here, and you see the gluten there, and a secondary signal. So ordinarily it doesn't bother people, although there could be sensitivities, but it doesn't result in an actual outright celiac. There's one thing interesting that they found, is that even though the